Good morning, everyone. Today we will start with meiosis. We will define meiosis. We will talk about the stages of meiosis, its importance in the production of gametes, which will unite to give the zygote, and the difference between mitosis and meiosis. First, mitosis, as we've taken in previous sessions, that we have a mother cell having 46 chromosomes or 2 N chromosomes. It will divide to give two daughter cells identical to each other and to the mother cell. Each cell has 2 N chromosomes or 46 chromosomes, but with one chromatid, unlike the mother cell here with two chromatids. But the number of chromosomes is conserved. It means the two daughter cells are identical to the mother cell. Mitosis is important in the growth and renewal of cells, as we've said. But meiosis is important in the production of gametes. Here in meiosis we have a mother cell that has 46 chromosomes or 2 N chromosomes. It divides to give two daughter cells, each having N chromosomes with two chromatids. Then each daughter cell divide into two daughter cells. Thus, at the end we have 4 daughter cells, each having N chromosomes with one chromatid. I mean here that we start with 46 chromosomes, here it becomes 23, and here 4 daughter cells, each having 23 chromosomes with one chromatid. It means the number in meiosis is reduced to half, from 46 to 23. This is because in gametes we need 23 chromosomes, because the gametes will unite 23 and 23 and will give the zygote 46. Thus we need meiosis for gamete production, and the 4 daughter cells are not identical to the mother cell. There is change in the genetic material. So this is the main difference between mitosis and meiosis. Here the number of chromosomes is conserved, 46 give 46, but in meiosis the number is not conserved, 46 gives each daughter cell having 23. As a definition, meiosis is a type of cell division in sexually reproducing organisms because meiosis occurs, as I've said, to produce gametes only and not in all body cells. It results in 4 daughter cells called gametes, each of which has half the number of chromosomes as compared to the original diploid cell. Diploid, it means, has 46, but the gametes are haploid, they have 23. The haploid cells become gametes, which by union by another haploid cell will give the new generation of diploid organisms by a process called fertilization. As I've said, that gametes will unite male and female gamete to give the zygote by fertilization. Meiosis occurs in the germ cells of sexually reproducing organisms. I mean by germ cells, which are the gonads, where it is testis in the male and ovaries in the female. These are the sexual cells where gametes are produced. Now we will talk about the phases of meiosis. We have two parts in meiosis: meiosis 1 or reductional division and meiosis 2 or equational division. In reductional division we have a reduction in the number of chromosomes from 46 to 23, and in equational we have the same number of chromosomes, 23 remains 23. We will start with the first meiotic division or meiosis 1, prophase 1. The first three figures refer to prophase 1. In prophase 1, it's somehow long in time because we have 3 parts we are going to talk about. First, the chromosomes are of 2 chromatids. Pairing up of homologous chromosomes. It means every 2 chromosomes that are similar or having same size, same position of centromere and same bending pattern, they sit next to each other. As we can see in figure 2, every 2 homologous 2 chromosomes that are similar, it means homologous chromosomes, they sit next to each other. Then we have a process occurring here called synapse. Synapse means every 2 chromosomes in the homologous pair, they synapse or come over each other, they exchange parts of the DNA. As we can see in figure 3, they come over each other. When they come over each other, we call this tetrad because they are now of 4 chromatids. They exchange parts of DNA. This leads to diversity in the gametes produced. Okay, here I want you to know that in prophase 1 we have pairing of homologous chromosomes and synapse. Okay, this is prophase 1. Now metaphase 1 in figure 4, they align at the equator forming the equatorial plate. Now anaphase 1, we have separation of homologous chromosomes. Here not sister chromatid at anaphase 1, the homologous chromosomes, it means those that pair up together, they will separate. Here the homologous chromosomes will separate and we will have two sets of chromosomes. In each set 3 chromosomes, as you can see. We started with 6 and now in the 2 daughter cells we'll have 3 by 3. Telophase 1, first cytokinesis or first division of the cell, we have constriction or cleavage furrow. We have 2 sets of chromosomes, 2 daughter cells, each having n chromosomes with 2 chromatids. It means I start with 6 and now 3 by 3. Each cell has 3, half the number of chromosomes. Now 7, we start with the second meiotic division. It is called here metaphase 2. There's no prophase 2. It means we start prophase 1, metaphase 1, anaphase 1, telophase 1, then directly metaphase 2. In metaphase 2, we have lining up of chromosomes at the equator forming equatorial plate, and we are working with 2 daughter cells because we have here 2 daughter cells. Then anaphase 2, we have separation of sister chromatids in each cell. It means now in anaphase 2 there is separation of sister chromatids. Sister chromatids separate in each cell, thus we will have at the telophase 2 4 daughter cells, because in each cell we have constriction of the membrane and we have 2 daughter cells. Thus the final result is 4 daughter cells, each having n chromosomes with 1 chromatid. Now I'm going to talk about changes of DNA concentration during the cell cycle. Cell cycle, I mean interphase plus mitosis or interphase plus meiosis. Interphase is important for the replication of DNA before mitosis or before meiosis. Starting with mitosis, first we have interphase before mitosis, here G1S and G2. In G1 the concentration here is 2N with 1 chromatid. Then we have S replication where the quantity of DNA doubles, thus we will obtain 2N chromosomes with 2 chromatids. It means 46 chromosomes with 2 chromatids. G2 phase, then prophase, then metaphase, the quantity remains 2N with 2 chromatids. Here the drop refers to anaphase 1 where there is separation of sister chromatids. Thus we will have 2N with 1 chromatid as we started here. Then we start another cycle G1 as G2. Okay, in the cell cycle plus meiosis, it means interphase plus meiosis, we have G1 as G2 interphase. It means starting 2N with 1, then 2N with 2 chromatids. Here it remains 2N with 2 chromatids. We start meiosis 1, prophase 1, metaphase 1. Then this drop refers to anaphase 1. We have separation of homologous chromosomes. The quantity drops because the number of chromosomes becomes half. It means I have now 23 chromosomes or n chromosomes with 2 chromatids. Then we have metaphase 2. Then this drop refers to anaphase 2. Anaphase 2 we have separation of sister chromatids. It means here it becomes half C because the quantity of DNA now becomes 23 chromosomes with 1 chromatid. This is the final result of meiosis. We have n chromosomes or 23 chromosomes with 1 chromatid, and then G1 phase or S phase starts again. So interphase comes before meiosis, not between meiosis 1 and 2. It comes before meiosis and before mitosis, and every cycle when we will start with every cycle starts with interphase, then meiosis or mitosis.